Hello, everyone. So we're just going to finish up with the remaining questions that we had. So we were in course test 2. And I'm going to finish up with the biology questions. All right, so we finished up with 34. Now we're going to do 43. Okay, so 43 asks us where would these amino acids lie if we were to put them into a gel electrophoresis, and the gel electrophoresis was buffered at pH of 6. Okay, so first what you have to know is that amino acids have different isoelectric points. So those are the points in which the amino acid will have charges, but they neutralize. For example, they have a positive charge or a negative charge, but those two things, they neutralize each other to make a net overall charge of 0, right? So the backbone of our amino acid is always going to be an amino group with a carboxylic acid group. Now let's say we are at a physiological pH. At a physiological pH, this carboxylic acid would be deprotonated, right? Because it's acidic, it's more acidic than the environment, so it's going to deprotonate. However, the amino group is not going to be deprotonated, so it's going to have all of its hydrogens. It's going to have a positive charge, and the carboxylic acid is going to have a negative charge. So overall, I have a net 0 charge on the molecule. That's your isoelectric point. Okay. So here in the question is asking us, okay, if we had a lysine, which is a base at physiological pH will have a positive charge. Glycine is a nonpolar molecule. Nonpolar molecules and polar molecules are going to have they're going to have a neutral charge at physiological pH. I know pH of 6 isn't necessarily physiological, but it's close enough to it that we can treat it as such, right? Around physiological pH would be like around 6, 7, or 8. At physiological pH is like 7.4, 7.3, but pHs 6, 7, and 8, you can just treat them the same thing as physiological pH. Yeah, so the mixture is composed of lysine, glycine, and aspartic acid. Aspartic acid is an acid. It's going to be deprotonated at physiological pH, which means it's going to have a negative charge, right? Just an overall negative charge, because they also have another carboxylic acid attached. Right, so they just have an overall negative charge for an acid at physiological pH or pH of 6. Okay, so basically saying, okay, what would be the order of these amino acids if they were put into this buffered solution in the gel electrophoresis? Well, the glycine, which has a net 0 charge, it wouldn't be attracted to a negatively charged terminal or positively charged terminal, so it'd be in the middle, right? It'd be 2. Lysine, the base with the positive charge, would be attracted to the negative terminal, so it'd be over here at the 1. And the aspartic acid, which would have a negative charge at this pH, it would have a negative charge and would be attracted to the positive terminal. So the correct answer would be in this order: lysine close to the negative terminal because it has a positive charge. Glycine, which doesn't have a positive or negative charge, would be in the middle. And aspartic acid, which has a negative charge, is going to be closer to the positive terminal. Okay. Now we're going to do question 51. Ooh, sorry. All right, so here we have the glycolysis pathway nice and drawn out for us. And our questions are going to be about this. So question 51 is just a memorization question. It's based on figure 1, which is the net reaction for the transformation of glucose to pyruvate. So guys, you got to know your pathways. Glycolysis is one of those pathways. Probably going to be one of the most asked about pathway. I would say the most asked about is the PPP, the glycolysis, and the Krebs of what I've seen so far. Electron transport chain as well. Okay, so basically we know that we're going to be taking glucose, we're going to oxidize it. We're going to be generating ATP, but in order to generate that ATP, we first have to sacrifice some ATP. And then later on, we're going to have something that is not ATP donate phosphates, right? Over here, the ATPs were donating phosphates. Now something that's called an inorganic phosphate is going to be attaching and adding phosphates to our intermediates, and then eventually create our ATP, right? Okay. And in this process, we see something like an arrow with a curved arrow, a straight arrow, and then an enzyme would be next to it. So this is the enzyme. The enzyme uses ATP to phosphorylate glucose to glucose 6-phosphate, and the ATP is used up, and we produce ADP plus hydrogen at the end. Okay, so let's see here. We're going to use up glucose. Here we use an ATP. Here we use an ATP. Okay. However, down here we produce an ATP, and over here we produce another ATP. So it seems like it'd be 0 ATP net, right? Like our product would be 0 ATP because we used up 2 and we also made 2. But remember that this phase of the glycolysis, it happens twice, right? Because we're going to have two 3 carbon groups go through this phase. So actually everything over here, you could just multiply by 2. Okay, so if we do all that, we'll find that we have a net of 1 glucose plus 2 inorganic phosphates plus 2 ADP plus 2 NAD pluses on the reactant side. And then net, we're going to produce 2 pyruvate, 2 ATP, 2 NADHs, 2 hydrogens, and then 2 water molecules. Whereas the water molecules at, it's over here. You just multiply this by 2, so it's 2 water molecules. The ATP, there's an ATP produced here, there's an ATP produced over here, and there is an ATP used up here, ATP used up there, but remember you multiply this pathway by 2, so it's 2 used up, but 4 produced, so net 2 ATP. We produce 2 pyruvate, and yeah, we just have to go through the graph, and honestly you should have this kind of stuff memorized, like what is being used, what is, what's it called, what is being produced for each of the pathways. Okay. Moving on. I'm going to do question 52. Same passage with glycolysis. All right, it says to produce the maximum number of ATPs, post-glycolytic pathways and aerobes should receive this, or must receive blah, blah, blah from glycolysis, right? And then we have the options. Okay, so let's break it down real quick. It says post-glycolytic pathways, right? So what are the pathways involved in cellular respiration, aka the production of ATP from the oxidation of foodstuffs? So first we have glycolysis, then we have pyruvate dehydrogenase complex, which produces the acetyl-CoA from the pyruvate produced from glycolysis. The acetyl-CoA goes into the Krebs cycle. The Krebs cycle oxidizes the acetyl-CoA until you get a bunch of NADHs, FADHs, and those things are electron carriers that donate electrons to the electron transport chain. The electrons then create a proton gradient. The proton gradient is then used to create ATP, right? Okay, so let's look at these options, right? So ADP and NADH. Well, NADH can produce ATP when it's put up in the ETC, but ADP is not going to produce ATP in particular. It's an intermediate, like it's needed when you're attaching the phosphate on it, but it's not necessarily going to produce an ATP. It would produce an ATP, but only one ATP, right? One ADP can be phosphorylated to create one ATP, right? So there's not a lot going on here. You guys remember NADH? It what's it called? It produces 2.5 ATPs per NADH. Okay, and then go to ATP and then another NADH. NADH does produce 2.5 units of ATP. And ATP in and of itself is not going to produce a lot of things. So, I mean, we talked about how ATP is used up in this process over here, but in terms of like post-glycolytic pathways, it's not going to be doing much. Like PDC, Krebs cycle, and ETC, these are the post-glycolytic pathways. And ATP is just not going to be cutting it in terms of like net overall huge maximum optimized use in the production of ATP. And here we have glucose and NADH. So glucose, when it oxidizes, can create a bunch of ATP, right? It's going to go through this entire pathway and eventually reach ETC. However, we're looking at post-glycolytic pathways, right? So the pyruvate dehydrogenase complex, Krebs, and ETC. So glucose is out for that reason. And next we have NADH and pyruvate, right? So NADH, yes, we said it produces 2.5 ATP each. And then we have pyruvate. And pyruvate is something that is going to be released from the glycolysis pathway, something produced in the glycolysis pathway, and it's going to be turned into acetyl-CoA through the pyruvate dehydrogenase complex, and acetyl-CoA becomes oxidized until it creates more NADHs, more FADHs that can pretty much create a super huge amount of ATP in the ETC. So pyruvate would be your best option. Okay, let's continue forward. All right, we're going to do question 53. All right, let me see here. Same passage. Okay, it says in glycolysis, how many moles of inorganic phosphate are consumed in oxidation of 3 moles of glucose to pyruvate? Okay, so what's glucose to pyruvate? Glucose to pyruvate is this process here. Where's the inorganic phosphates used up in this pathway? ATP, is that in inorganic phosphate? No. An inorganic phosphate is something that looks like this. It's just a phosphate group, like this is a phosphate group, and usually it's held as a salt, right? A negative charge with a positive charge as a salt, an ionic bond. This would be an example of an inorganic phosphate. Like here's ADP, adenine diphosphate, and an inorganic phosphate is just going to attach, and it's going to make ATP. So we're not looking for we're not looking for a phosphate group that is donating. We're not looking for a phosphate group that is donating to the glycolysis pathway that's attached to something organic. So adenine has a bunch of carbons. That's an organic molecule. Is there any carbons here? No, right? Just a bunch of phosphates, oxygens, and hydrogens. So that's not organic. If it's not organic, it's inorganic. So where are the inorganic phosphates at? We scroll down and we find it here. Remember, this pathway happens twice. So whenever we try to create pyruvate from glucose, we're going to be producing, or sorry, we're going to be using 2 inorganic phosphates, right? Because remember, this pathway goes down twice. So you just multiply this one phosphate by 2. So it's 2 phosphates per glycolysis pathway. And here it says we have 3 moles of glucose to turn to pyruvate. 3 moles of glucose is just saying like, okay, like yeah, that's just a quantity, right? So how many moles would we need of inorganic phosphate? We said that we needed 2 per glucose. So we just multiply 3 times 2 and we get 6. Okay, next question. Yeah, I was gonna say like what moles is, but I'm not entirely sure. I think moles is just a quantity of something with Avogadro's number. Could be wrong, but I kind of forgot. All right, so here we have a, what's it called? A freestanding question. All right, so it says retinol isomerase is used to alter the conformation of retinol. Okay, cool, nice little fact. Retinol isomerase is most likely manufactured by, wait, is that enough information? I'm not sure. What do we know about retinol isomerase, right? It's asking us, how is it manufactured? Well, retinol isomerase, if you see this tag at the end, this suffix ase, it means that it's an enzyme. And enzymes are all proteins, or most enzymes are proteins. There are ribozymes. But if they don't specify that it's not a ribozyme, then you have a protein structure. And protein structures, just like the other macromolecules, you have what's it called, lipids, proteins, nucleic acids, and carbohydrates. They're all produced via this process called dehydration synthesis. And it's basically what it sounds like. Dehydration is the kind of like, it's water related, right? Hydration is water. Dehydration is the removal of water. So whenever you have a macromolecule, what happens is that there's a structure here, there's an OH, another structure here that has an H. And what happens is that you snatch away an OH and an H from those molecules, and then you, what's it called? And then you produce an H2O. So yeah, that's dehydration synthesis, the snatching of an OH and an H, and you're producing H2O right there. So that's the correct answer. This is really just a terminology kind of deal. Like, do you know what dehydration synthesis is? Yes. Can you tell that retinol isomerase is a protein that due to the fact that it's an enzyme? Yes. And then you can answer it. Let's look at the other options. This is hydrolytic synthesis. Hydrolytic synthesis, not really. Hydrolysis is usually something that you use to cut things. Like, for example, we created proteins, carbohydrates, and nucleotides and lipids with dehydration. The things that cut them, like lipases or proteases or nucleases, or for carbohydrates, it's amylases. Those things usually add a water molecule to reform these groups so they can separate monomers. So hydrolytic synthesis is kind of like a weird term here. It's kind of like destruction, create. It's kind of odd. Oxidation, no. And glycosidic anabolism. Well, remember, retinol isomerase is a protein, not a glyco. A glyco, it means sugar. So yeah, you can cross that out for that reason. Okay, lastly, let's hit up 58. Okay, it's another freestanding, and it says glucose 6-phosphate dehydrogenase deficiency is an inheritable metabolic disorder that can result in the destruction of red blood cells, and in severe cases can lead to kidney and liver failure. Which of the following best describes the reason why G6PD deficiency can result in premature red blood cell destruction? Okay, so destruction, right? We need to find that something that's going wrong. Okay, but first, what does G6PDH do? So this is an enzyme that's found in the pentose phosphate pathway, and in particular in the particularly in the oxidative branch of the pentose phosphate pathway. There's the non-oxidative branch, the non-ox, which produces glycolytic intermediates or just like sugars. And then we have the ox branch, which produces NADPH plus ribose 5-phosphate. Ribose 5-phosphate is basically the backbone of nucleotides, phosphate group attached to a ribose sugar. And NADPH is a super duper reducing agent, right? It's not going to be used as an electron carrier like its cousin, the NADH. NADPH is a super reducer, so it's really used for the reduction of things. And in the pentose phosphate pathway and the oxidative branch, glucose 6 glucose 6 phosphate dehydrogenase enzyme, what it does is that it produces NADPH. Okay, so if we have a loss of this enzyme, that means that we have a loss of this thing. So what does a loss of this thing really do? Like, how does that hurt? Well, for one thing, what happens is that NADPH, since it's a super reducer, what it does is that it rejuvenates something. It rejuvenates something by reducing it. So there's something called glutathione. Glutathione. Thione. I hope I spelled that correctly. I think I have it in the glutathione. Yes. Okay. So glutathione is a species that acts to combat ROS. ROS is reactive oxidative species. These reactive oxidative species means oxygen. They're produced in the electron transport chain sometimes when the electrons that come from the ETC. So ETC is like this. You have a bunch of electrons come down, and the final electron acceptor is O2, right? So sometimes you get an odd number of electrons attached to your oxygen, and what happens is that you create a radical. So here we have a pair here, pair here, pair here, but this electron is not paired, and this is really dangerous because it's you have a radical that really likes to be paired up with something, so it's really reactive. So imagine this species is ROS, this reactive oxidative species. If it were to enter the nucleus, it would damage a lot of your nucleotides. And what the cell does, if it's not able to like kind of repair any damage to itself, the cell just decides to do apoptosis, right? It's like, okay, I'm going to die, screw it. Like I don't want to make anything bad. I don't want to be a bad cell. I'd rather just kind of like do apoptosis and kind of like eliminate the threat before it becomes something serious, right? And so if glutathione can't be rejuvenated by NADPH, so what glutathione does is that it reduces these oxidative species, so it can no longer be reactive. It can no longer be a radical. But this uses up the reductive power of glutathione. And glutathione is rejuvenated by NADPH. So if we were to have this kind of disease where we have a deficiency of this enzyme, we can't have NADPH. If we don't have NADPH, we can't have glutathione rejuvenated. If glutathione is not working, we have these oxidative species that can really mess up a cell, causing it to pretty much have a premature red blood cell destruction, right? It's not happening. The cell decides to kind of like stop the threat before it becomes worse. And the correct answer for that reason would be C. Glucose 6-phosphate dehydrogenase deficiency results in the deficiency of NADPH, which results in the increased damage from reactive oxidative species and causes premature cell death. And why does it mention the kidney and liver? Because these are kind of like filtration slash like organs that deal with the blood. And so they have a lot of blood if you have something malicious in the blood, like contents of the red blood cell, like if a red blood cell were to be destroyed, a lot of its contents need to be filtered out and stuff. So you would have a lot of these things, a lot of these organs kind of trying to deal with that, and they can get hurt. Okay, last question, question 59. And you guys remember you have all these PowerPoints with you in the Sakai. Okay, so yes, we have a lab technique here. It's called extraction. Basically, what extraction is, it's the kind of removal of something from a mixture, right? Let's say we have a mixture here, and we have something that looks like this and something that looks like that, right? And we want to separate these two things, but they're both dissolved in this mixture, right? So what extraction does, this process, this lab technique extraction, is that it takes advantage of the fact that things are likely to be more soluble in a particular medium compared to a different medium. And so what happens is that you start off with your mixture generally in an organic solvent. So an organic solvent is something that is going to dissolve things that are going to be, in particular, non-polar. That's just what's designated as the organic solvent. They dissolve non-polar things. And what you're going to do is you're going to manipulate one of these things. To say this guy, you're going to manipulate this guy so that it can no longer be, less soluble in organic phase, but rather be more soluble in a different phase. Let's say a polar phase, right? Something that is polar like water, right? So what you do is that you first manipulate this here substrate, or like this species, and then you add water, H2O, and then you mix them together. And what happens is that you're going to have two different phases, one phase over here, A, and one phase over here, B. And so now this thing that's once used to be able to dissolve in the organic phase is now more soluble in the polar phase, and now you've effectively separated the two things. This is the organic phase, this is the polar phase, and all you would have to do now is just kind of like separate these two phases, right? Like they're separated by density. The water happens to be more dense than the organic phase, so you just drain this part out, and then this thing is left behind. Okay, so the question is asking us like, hey, like let's separate these two things. How can you do that? Well, guys, remember, if something has a charge on it, it's most likely going to be attracted to dipoles. Remember, dipoles are kind of just like an uneven distribution of an electron cloud. For example, in water, water is a really good polar solvent because the oxygen is so electronegative that it pushes all the electron density to itself, making it partially negative charge, and the hydrogens are going to have a partial positive charge. So if you were to have if you were to throw on a charge onto, let's say, benzoic acid, if you were to throw on a charge onto this benzoic acid, then it would be really attracted to a polar solvent like water. Okay, so how can we give a charge to benzoic acid, and we're trying to separate it from aldehyde? Well, benzoic acid is an acid, which means that it can donate its proton. It can leave its proton. If the proton dips, then the covalent bond between the oxygen and the hydrogen, those electrons in that covalent bond will go to the oxygen, giving oxygen an overall negative charge. And that's how you can separate the two, right? So you give a charge to benzoic acid by, let's say, removing that hydrogen with a base, and then you have a charge on this guy, you introduce it to water, and now it dissolves into the water, and then you can extract it, right? So why won't a base affect this benzaldehyde? Well, this hydrogen here isn't acidic, right? It's not really that acidic. This benzoic acid, this hydrogen over here, it is acidic, so it can leave. So basically, it's asking us, hey, like what can we use for extraction? Like what here is a base that can remove this hydrogen? This is right here bicarbonate. This can act as a base. Right, the Na is just a counter ion. So if you were to draw it out, it's Na plus HCO3 minus. So this can be effectively a base. And then HCl, that's a strong acid, that's not going to help us. And then NaOH is a strong base that can definitely deprotonate benzoic acid. So the correct answer, 1 and 3. Okay, guys, that'll be it. I'll be uploading this to YouTube soon, and I'll see you guys on Saturday.